welcome to today's video lecture. In today's video lecture, I'll be solving previous year's NEET questions and discussing the answers. And for the chapter Biotechnology and its applications, and we are in part 2 of the series. So if you haven't checked out part 1, please do so. The link will be there in the description box below. All right, so let's begin. Okay, so question number 1. The bacterium Bacillus thuringiensis is widely used in contemporary biology as, and this appeared in 2009, A. Insecticide, B. Agent for production of dairy products, C. Source of industrial enzyme, and D. Indicator of water pollution. Okay, so Bacillus thuringiensis is widely used in contemporary biology as an insecticide. Right? We've already discussed the function of Bt toxin. Okay, so the answer is A. Let's go to the discussion. So Bt cotton contain toxin for insecticides obtained from Bacillus thuringiensis. There's Bt maize also. Okay, so there are there's an endotoxin that's produced. I'll just explain it quickly again in brief, but please check out part 1. I've explained this in detail. So Bacillus thuringiensis is a bacteria, okay, and it has a gene called cry gene which encodes for a protein which is toxic to insects. Okay, now there are some genes that are useful in the cotton bollworm. Okay, so it behaves like an insecticide in cotton bollworm. That is I think cry1 AC and cry2 AB. These two genes are behave like an insecticide for cotton bollworm. Okay, so cotton bollworm is an insect which destroys the cotton plant. So now if we incorporate these two genes in the cotton plant, then it will be able to produce a toxic protein that will kill these insects. Okay, so that's why this is useful. Now, how does it kill the insect? So the toxin, once ingested by the insect, is activated by the alkaline gut. So now the gene encodes for that protein, right? So when the toxic protein, it does not destroy the plant or the bacteria that is producing it also. So it's a very unique type of protein, toxic protein. It goes into the insect gut. The insect gut is alkaline in nature, and there it converts into the activated form of toxin, and that's how it then kills the insect. Okay, so how it does it binds to the surface of the midgut epithelial cells and creates pores that cause swelling and lysis. Okay, so this is how the toxin kills the insect. All right, clear? I think I've answered the question though. Yeah. Okay, let's move on then. Question number 2. What is antisense technology? Okay, this appeared in 2009. A. When a piece of RNA that is complementary in sequence is used to stop expression of a specific gene. B. RNA polymerase producing DNA. C. A cell displaying a foreign antigen used for synthesis of antigens. D. Production of soma clonal variants in tissue culture. So antisense technology is A. When a piece of RNA that is complementary in sequence is used to stop expression of a specific gene. Okay, so the answer is A. Let's go to the discussion. So in the previous lecture, I spoke about RNA interference or RNA silencing, right? It's the similar concept here. Let's say you know that DNA is transcribed to mRNA, right? And mRNA then translates to proteins. Okay, we know that. But let's say you have the mRNA, but you don't want it to express the protein. Okay, we don't want that. So what we do is we stop the expression of protein by introducing a complementary strand of mRNA. Okay, so let's say if I introduce a complementary strand of mRNA, then once this binds with this mRNA, it will stop the expression of protein. Now mRNA cannot be translated into protein. All right, so it obstructs the translation into a protein. So basically that's what antisense technology is. It's a similar concept in RNA interference, RNA silencing. Let's go to the discussion. I'm sorry, the question. So yeah, when a piece of RNA that's complementary in sequence is used to stop expression of a specific gene. Clear? Let's move on. Okay, so question number 3. Cryo-1 endotoxins obtained from Bacillus thuringiensis are effective against, this appeared in 2008, nematodes, B bollworms, C mosquitoes, D flies. So I already discussed this again. So they are endotoxins obtained from BT, Bacillus thuringiensis, effective against bollworms. Okay, bollworms are insects. So it is B. So you have a corn bollworm, you have that affect the cotton plants, you have corn bollworm that affect the corn plants, the maize plants also. So they are, so their cry1 gene is also a gene, a type of gene that is present in Bacillus thuringiensis that encodes for this crytoxic protein. All right, clear? I think I answered the question. Can move on. Yeah, okay. Cry1 AC and Cry2 AB controls the cotton bollworms. We discussed this also. Okay, let's move on. Question number 4. A transgenic food crop which may help in solving the problem of night blindness in developing countries is, this appeared in 2008, A. BT soybean, B. golden rice, C. flavor saver tomatoes, and D. Starlink maize. Okay, so the question is a transgenic food crop that will solve the problem of night blindness, and that is B. golden rice. Okay, so the answer is B. I already discussed golden rice also. It's a genetically modified crop. It was created for the synthesis of beta carotene. So once we consume beta carotene, it gets converted to retinol, which is a form of vitamin A, and that cures night blindness. Okay, so night blindness is an indication of vitamin A deficiency, especially in developing countries where there is no access to proper nutrition. These genetically modified crops help in countering that deficiency. So yeah, we know that beta carotene is a precursor for the formation of vitamin A. Okay, and oops, yeah, sorry, it was wrong order, but yeah, it contains two genes, PS1 and CRT1. Okay, already discussed this also in the previous lecture. But to answer your question, we've answered the question. It's pretty direct, simple. Let's move on. Question number 5. Main objective of production or use of herbicide resistant GM crops is to, this appeared in 2008, A. Encourage eco-friendly herbicides, B. Reduce herbicide accumulation food articles for health safety, C. Eliminate weeds from the field without the use of manual labor, D. Eliminate weeds from the field without the use of herbicides. So the main objective of using this production of herbicide resistant GM crops was to reduce herbicide accumulation in food articles for health safety. Answer is B. Now herbicide is useful for weed management, we know that, right? But it also affects the plant, the crop that is there. So they're useful in managing growth of weeds, but it also harms the plants, causing diseases to it. Okay, so hence GM resistant, herbicide resistant crops were formed or created. Okay, to protect the plants from the harmful effects of herbicides. All right, so this is one of the reasons. All right, let's move on. Question number 6. Golden rice is a promising transgenic crop. When released for cultivation, it will help in, this appeared in 2006, A. Producing a petrol-like fuel from rice, B. Alleviation of vitamin A deficiency, C. Pest resistance, D. Herbicide tolerance. Again, it's the same thing. It alleviates vitamin A deficiency. Answer is B. So genetically modified crop golden rice was created for the synthesis of beta carotene, and beta carotene is a precursor for the formation of vitamin A. It's the same answer. All right. So basically golden rice, yes, was formed to alleviate the deficiency of vitamin A. All right, let's move on. So simple. Question number 7. Bacillus thuringiensis strains have been used for designing novel This appeared in 2005. A. Biofertilizer. B. Biometallurgical techniques. C. Biomineralization processes. D. Bioinsecticidal plants. So it is clearly D. Bioinsecticidal plants. Okay, so answer is D. Again, it's the same explanation. I'm not going to go into the details. You can watch my previous lecture or the answer just before that. I think a few questions before that I've explained it. Yep, so this is it. Let's move on. Question number 8. Golden rice is transgenic crop of the future with the following improved trait. This appeared in 2005. A. Insect resistant. B. High lysine essential amino acid content. C. High protein content. D. High vitamin A content. So the golden rice transgenic crop was produced to counter the deficiency of vitamin A, so to produce high vitamin A content. So the answer is D. Again, golden rice. I think BT and golden rice have been repeated numerous times. Oops, 1 second. So yeah, I've already explained this, so I don't want to repeat myself again. You can either take this down or watch my previous explanation. Okay. Question number 9. The first transgenic crop was, and this appeared in 99. A. Tobacco. B. Cotton. C. Pea. D. Flax. So the first transgenic crop was A. Tobacco. Okay, learn it as a fact if they ever ask you. 1 second. The first transgenic plant, actually should have been, was tobacco. All right, learn it as a fact. Let's move on. Question number 10. Match the following columns and select the correct option. This appeared in NEET 2020, pretty recent. A. BT cotton would be Bacillus thuringiensis. Adenosine deaminase deficiency is gene therapy. RNAi is cellular defense. Sorry, RNAi cellular defense. PCR is detection of HIV infection. So we have A4. Okay, that's the first one. A4, B1, C2, and D3. Okay, so the answer is A. Let's go to the discussion. So BT cotton is associated with Bacillus thuringiensis. We already know the connection between the two. Now, adenosine deaminase deficiency. Okay, now this was the first, the gene therapy was done on a 4-year-old girl in 1990. Okay, now sometimes there are babies born that have adenosine deaminase deficiency, and that is not useful for the human body because it affects the immune system. Okay, so if somebody has an ADA deficiency, that means they will eventually counter SCID, that's severe combined immunodeficiency. Okay, and basically ADA plays an important role in developing the immune system. Now how is that? Okay, so ADA, adenosine deaminase, okay, breaks down. I'm sorry, when DNA breaks down, it forms deoxyadenosine. Okay, now when DNA breaks down, it forms deoxyadenosine. And deoxyadenosine is harmful or toxic to lymphocytes. Okay, it's toxic to lymphocytes. Now, so basically we don't want deoxyadenosine around, right? So adenosine deaminase, ADA, converts this into a less toxic form. Okay, converts this into a less toxic form. So that's the role of ADA. So just imagine if there was ADA deficiency, we got a gene that did not produce ADA, that defective gene was present in the person. So obviously it would destroy all the lymphocytes, and then that would then affect the immune system of the person, right? So what scientists developed is a way to counter that problem, okay, by providing ADA to the person. Now how they did is a permanent, not a permanent way I would say, but after the person is born, the baby is born and it's grown up, so that's why it was done on a 4-year-old girl. there was a method by gene therapy, okay, by which they could introduce the gene into the person. So what they did is the gene coding for ADA was inserted in her using retroviral vectors. Now we know that what they created basically was rDNA. Okay, so they used retroviral vectors. They introduced the ADA gene in it and then put it into the patient. Okay, hosted by the patient's lymphocyte. The host was the lymphocyte, and this was done via periodic infusions. Okay, this was done via periodic infusion. That means over a period of time they would just keep continuously inserting those vectors in so that the vectors would produce the protein. Okay, so that's how they could counter the ADA deficiency. We will come to this later on in the future questions also. They have asked this over and over again, but this is just to give you a brief on what ADA does, okay, and how ADA deficiency was countered by rDNA technology. Now, RNA silencing also we want to discuss was found to naturally occur in eukaryotic organisms as a form of cellular defense. And PCR is used to amplify DNA and it's used to detect pathogens. So if you go back, yeah, we've answered the question. We can move on. Okay, so question number 11. Which of the following statements is not correct? This appeared in NEET 2020. A. In man, insulin is synthesized as a proinsulin. B. Proinsulin has an extra peptide called C peptide. C. The functional insulin has A and B chains linked together by hydrogen bonds. D. Genetically engineered insulin is produced in E. coli. So to find the incorrect statement, the one that's not correct, and that is C. The functional insulin has A and B chains linked together by hydrogen bonds. That is incorrect. Okay, so the answer is C. So here you have a typical structure of insulin. You have proinsulin, which has A, B, and the C peptide, right? So now the mature insulin, so basically initially it's produced as proinsulin, then it gets converted to mature insulin. The mature insulin has A and B peptide. It does not have the C peptide. All right, and they are bound by disulfide bridges. Okay, so functional insulin A and B chains are linked together by disulfide bridges. And this is taken from NCERT, so you should know this diagram. Okay, so this is basically how proinsulin is converted to insulin, the mature form that we use. Okay, but to answer your question, yeah, the rest A, B, and D are correct. C is the incorrect statement. Okay. Okay, so question number 12. Which kind of therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency? This appeared in NEET 2016. We already discussed this briefly, right? So, yeah, it's A, gene therapy, B, chemotherapy, C, immunotherapy, D, radiation therapy. The correct answer is A, gene therapy. All right, answer is A. So the first gene therapy was done on a 4-year-old girl in 1990 who had adenosine deaminase deficiency leading to SCID. Already discussed how adenosine deaminase deficiency affects the person. Okay, it affects the immune system because when DNA breaks down into deoxyadenosine, it is very toxic to lymphocytes, and so adenosine deaminase converts it into a less toxic form. So just imagine if there was deficiency of ADA, then it would affect the lymphocytes and then eventually affect the immune system. All right, so what they did is they used retroviral vectors in which they introduced that into the person, the patient, and then that retroviral vectors eventually produce the ADA protein, the enzyme. Okay, so but that was done via periodic infusion. That means it was given over and over again. Okay. But to answer your question, yes, it was done via gene therapy. So that's how gene therapy, the discovery and the invention of this method, this therapy proved to be very useful. So yeah, gene coding for ADA was inserted in her using retroviral vectors hosted by the patient's lymphocytes. So the host was lymphocytes. Okay, the host was lymphocyte, and this was done via periodic infusions. Okay, clear? Let's move on. Question number 13. The two polypeptides of human insulin are linked together by, this appeared in NEET 2016, A. Covalent bond, B. Disulfide bridges, C. Hydrogen bonds, D. Phosphodiester bond. We just discussed this few questions back. So the two polypeptides of human insulin are linked together by disulfide bridges. Answer is B. Okay, so you have your A chain and you have the B chain here, and you have this disulfide bridges. Okay, that's how these polypeptide chains are linked to each other. Okay, let's move on. Question number 14. The first human hormone produced by rDNA technology is, and this appeared in 2014, A. Insulin, B. Estrogen, C. Thyroxine, and D. Progesterone. So the first human hormone produced by rDNA technology was A. Insulin. Answer is A. So there was an American company called Eli Lilly Biotechnology company, and they managed to do that in the year 1983. Okay, so what they did is they prepared two DNA sequences corresponding to A and B. Okay, they were the chains of the human insulin, right? And introduced them in plasmids of E. coli to produce insulin chain. So the E. coli was the host. Okay, so they produced A chain separately and they produced B chain separately. Okay, and then they were produced, yeah, of course they were produced separately, extracted and combined by creating disulfide bonds to form human insulin. So for those patients who cannot produce this insulin in their body because their pancreas is not functioning, this insulin proved to be very useful to them, right? So in 1983 the company was able to do this. Okay, and again this is another example of how sorry rDNA technology proved to be a very useful tool in medical technology. First we saw gene therapy, then we saw the production of insulin. Yeah. Oh yeah, insulin was the first human hormone. Okay, let's move on. Question number 15. Which one of the following vectors is used to replace a defective gene in gene therapy? This appeared in NEET 2013. A. Adenovirus, B. Cosmid, C. RI plasmid, and D. TI plasmid. Now, in gene therapy, we have seen retroviral vectors play a role, right? When we saw how ADA gene deficiency was fulfilled. But there's another type of vector called adenovirus vector. So the option is A. That is also used in gene therapy. They also use in gene therapy. It is a non-enveloped virus and has double-stranded DNA genome. Okay, so just like retroviral vectors, adenoviral viral vectors also play a role in gene therapy. Okay, there's another option called RI plasmid in the section, the option section, and it's found in agrobacterium. I thought I'll just mention it. It's found in agrobacterium rhizogenes and causes hairy root diseases in dicot plants. We know about TI plasmid, right? So there's another plasmid called RI plasmid also, right? TI plasmid we already know, cosmid we already know. So yep, this answers our question. Let's move on. Question number 16. The first clinical gene therapy was given for treating, this appeared in means 2012. We already discussed this also. A. Diabetes mellitus, B. Chickenpox, C. Rheumatoid arthritis, and D. Adenosine deaminase deficiency. So it is D. That's the first clinical gene therapy that was done. Answer is D. Okay, so I'm not going to go into the details again. I already discussed this. You can go back and listen to my discussion. Right, let's move on. Question number 17. Which one of the following is now being commercially produced by biotechnological procedures? This appeared in mains 2010. A. Nicotine, B. Morphine, C. Quinine, and D. Insulin. So insulin is now being commercially produced by biotechnological procedures. Answer is D. Okay, so that also again how Eli Lilly, an American biotechnology company, in 1983 managed to produce insulin and commercially is now also sold. And yeah, that was a revolutionary, achievement in terms of medical treating medical diseases, and it helped patients that could not produce insulin in their body. So this artificial insulin production helped them counter that insulin deficiency in them. All right, clear? You answer the question. Let's move on. Question number 18. The genetic defect ADA deficiency may be cured permanently by, this appeared in 2009. A. Administering ADA activators. B. Introducing bone marrow cells producing ADA into cells at early embryonic stages. C. Enzyme replacement therapy. D. Periodic infusion of genetically engineered lymphocytes having functional ADA cDNA. Now, it says may be cured permanently. Okay, so there's one way that you can do it, and that is option B. We know that periodic infusion can be given to somebody who's a little older, right? That happened to a 4-year-old girl. But that's not like a permanent solution. Let's say if you want to find a permanent solution to it, then you could use this technique. Okay, that's option B. We'll come to this. So the answer is B. So permanent way of solving the ADA deficiency problem would be to isolate the gene. Let's say you have the ADA gene with you, and in the embryonic stage insert it. Okay, and in the embryonic stage inserted during the early embryonic stages. Okay, so that is one way of doing it. So you isolate the gene from the bone marrow cells and insert it to the fetus during the early embryonic stages. So when it's in the embryo form, that's one way of solving this problem permanently. Sorry. So to answer your question, if we go back, yeah, so D is what the gene therapy was able to do using retroviral vectors and so on. But that was for a person who's a little older. But in the case of permanently curing it, there is a way to do it, and that could be option B, by introducing the bone marrow cells producing ADA into cells at early embryonic stages. Okay. Right, let's move on. Question number 19. Human insulin is being commercially produced from a transgenic species of, this appeared in 2008, Rhizobium saccharomyces, C. Escherichia, D. Mycobacterium. So human insulin is produced from a transgenic species of C. E. coli. Okay, so yeah, so what they did is they we discussed this. They created the rDNA, introduced into the E. coli. Yeah, and then this E. coli was able to produce the A chain and the B chain. And so this A chain and B chain were then bound together by disulfide bridges. Okay, and that's how they were able to then commercially sell this human insulin, because the insulin is used in the A chain and B chain form, not in the pro-insulin form, right? So we needed something that was already in the mature form and then put into the body. Okay. I guess I've answered the question. Yep. Question number 20. ELISA is used to detect viruses where the key reagent is, this appeared in 2003 and 2004. A. Alkaline phosphatase, B. Catalase, C. DNA probe, D. RNase. So it's used to detect viruses where the key reagent is A. Alkaline phosphatase. Answer is A. Now to understand this, we need to understand what ELISA is, right? So ELISA is enzyme linked immunosorbent assay. Okay, that's the full form of ELISA. So it's an acronym. So it's based on the immunochemical antigen antibody reaction. We know antigen antibody reaction that happens in the immune system, right? So the same concept is applied in ELISA. So pathogens such as viruses, bacteria can be detected. Even hormones, cytokines can be detected by this method. Now what happens is, let me just briefly explain what actually happens in ELISA. So let's say you have a plate here. Now there are two types of antibody that are used. There's a primary antibody and there's a secondary antibody. Let's say you have a plate over here and you have primary antibody attached to the plate. Okay. Now, and you have your, let's say you have your pathogen, your virus or your bacteria that you want to detect. Okay. So you have your, the bacteria, when you put it on the plate, it will attach to this primary antibody. All right. Then you have a secondary antibody. A secondary antibody is then placed on the plate. Now this secondary antibody is connected to an enzyme. It's an enzyme linked secondary antibody. It's an enzyme linked secondary antibody, and this enzyme is in this case alkaline phosphatase, let's say. It could be peroxidase also. Peroxidase is also used. In this case, it's alkaline phosphatase. Okay. Now, once the secondary antibody with the enzyme linked is attached to this pathogen, the antigen, let's call it the antigen. Now, you add a substrate. You add a substrate. Now, once you add the substrate, a substrate enzyme reaction will begin to happen here. Okay, substrate enzyme reaction begins to happen here, and you will see a change in the color. Okay, you will see a change in the color, and from the intensity of the color, you can determine what is the concentration of the pathogen, the bacteria or the virus or the hormone or the cytokine that you're trying to detect is present in that particular sample. Now this ELISA is usually used in labs for detecting, like I said, viruses, bacteria, the amount of hormones in a particular sample. So if a blood sample is given to you, let's say, and you want to detect the amount of estrogen or the progesterone or testosterone in that particular sample. Okay, so you don't usually use blood directly. What you do is you extract the serum from it. So you centrifuge the blood and you get the yellow form. I hope you know what serum is, right? You have the blood at the bottom and you have the, let's go back. You have the yellow color serum on top. So you take out the serum and that is your sample. Okay, that is your sample. You could even use urine. There are some commercially available kits that also say you can use serum, you could use plasma, you could use urine samples. Let's say you're using serum. Okay, you're using serum, and that serum contains the amount of estrogen or the progesterone or whatever it is, but you don't know how much of it is there. Then there are commercially available kits which are designed in such a way to help you determine the concentration of that particular hormone, let's say estrogen, and then you perform the protocol that is there in the kit, which is this protocol that I've just mentioned here. So you have a primary antibody that is plated on the plate. You get a 96 well plate. Okay, there's a 96 well plate over here. Imagine there are 96 wells. There are 8 columns. There are 12 columns and there are 8 rows. 3, 4, it goes on till 8, and there are 12 rows, 12 columns. Okay, there are 12 columns and 8 rows, and in that you have to put in your sample and you have to perform the protocol. Like I just mentioned here, you have your primary antibody already there, you put in your serum samples there, then you put in the secondary antibody which is linked to an enzyme, and you add the substrate. Okay, and then you see the change in the color, and that intensity of the color will determine how much of concentration of that particular thing that you're trying to detect is there. Okay, now that is measured using a spectrophotometer. Okay, spectrophotometer is a machine which determines the intensity of that color, okay, with the help of the wavelength of the light. Okay, that is also mentioned in the kit, if it's at 450 nanometers or whatever it is, that case, whatever the case is there. Special mention, you have your unknown samples. Okay, it has you've already done your experiment, you've put it in the spectrophotometer, you've got some reading. Okay, it gives you a reading in the form of optical density, OD, optical density. I'm sure you'll learn all this when you go to college, when you do your practicals over there, to determine the amount of protein or whatever. So you get the reading in terms of optical density. Now what the hell is optical density, and how do you measure that against your sample? How do you know what is the concentration of the sample, right? So the sample concentration should usually be nanograms per ml or picograms per ml or whatever that kit has mentioned to you according to their protocol. So there is something called a standard graph. There's something called a standard graph that needs to be um, measured, okay, before you can do this unknown sample. Now how do you get the standard graph? There are standards mentioned or given to you by the commercial kit that you're using. Now the standard graph is designed in such a way that you already know the concentration of particular standards. Let's say, for example, and just erase this. I hope you've understood this part. I've already explained the part where you can find out the how to use the unknown sample. You don't know the concentration, but you have to find the concentration. How do you find the concentration of that? By using standards. Now standards are set concentrations. Okay, let's say you already given a sample which is already nanograms per 0 nanograms per ml. You have 20 nanograms per ml, you have 40 nanograms per ml. You already have the sample with you, have 80 nanograms per ml, 100 nanograms per ml, okay, 120 nanograms per ml. Sorry. So you already have the set concentrations with you already. Okay, you do the same experiment on that. Okay, same experiment on that, you will get the optical density of that, but you already know the concentration. You have 80, you have 100, you have 120. Okay, you have the optical density. Let's say 0 will be, let's say 0, it will measure something, I don't know, 0.1, 0.4, 0.8, I'm sorry, 1.2. You have the optical density here, and you have your set. So we know that at 20 you should get 0.4, at 40 or you should get this, at 80 you should get so and so. So a plot is made, a graph is made. Let's say you get a straight line graph. You're doing some experiment and your result is a straight line graph here. Okay, it's a straight line graph here. Usually when you're using standards and you measure that in a spectrophotometer, you get a particular reading, right? Usually the kit will mention what kind of graph you're supposed to get. It's a straight line graph, or it's a curve graph, or so on. But for our purpose, let's say we get a straight line graph at the end. Okay, at the end of the experiment of the standards. Now we have this particular graph with us. We have already measured the optical density of the unknown samples. Okay, we got something, we got, let's say, 0.5. Now 0.5 should be somewhere here, let's say. Using a graph paper, please, okay? Don't do it as I'm doing it. Let's say you get it as, you get your optical density at 0.5. So, you know, your standard graph is fine because it has measured exactly as per as per as per the manufacturer has mentioned in the kit, okay, that 20 should be over here and 40 should be here and 80 should be here. And yes, you got the correct readings and you got your straight line graph, which is very good. Now next is 0.5. Let's say you got your unknown sample at 0.5. Unknown sample is 0.5. Okay, so you determine that 0.5 here and you get, I don't know, 25 or 28, something like that. You get a reading at 28 nanograms per ml. So you know that unknown sample has a 20 nanograms per ml of, I don't know, estrogen or whatever it is that you're measuring. Okay, so the standard graph is also important in ELISA when you are measuring unknown samples. Now this is basically the overall method of how ELISA is actually done in a lab. Okay, that's how ELISA is actually done in a lab, and that's how you get the answer. Obviously this is very briefly explained in NCRT and, you know, other books, but this is actually how it's done in a lab. First you need your standards set. Standards, you know, there are fixed concentrations. For that you get your optical density and you will get a graph, with a straight line, it could be a curved graph, whatever the case is. Okay, so that is your fixed standard graph. All right, you can't get a graph that goes like this and then comes down again. No, that is clearly incorrect. Okay, either it will be a straight line graph or it will be a curved graph. Okay, so once that is done, once you got a standard graph set, then your next is to find the concentration of the unknown sample. Okay, now you have your unknown sample. Let's say you got it as 0.5, or you got your optical density at 0.6. Optical density is basically a number that is given to you by the spectrophotometer after it has measured the intensity of that color. Right, when the substrate enzyme reaction takes place, you'll see a change in the color. So depending on how much of the sample is present, I'm sorry, the antigen is present in the sample, it will change the color accordingly. Okay, so if there's too much of let's say estrogen in a particular sample, the color will be very dark, but if there's less estrogen in the sample, the color will be very light. So obviously you can't just see it with your bare eyes. It's impossible to determine that. So a spectrophotometer does the job for you, and it will give you a reading. It will say the optical density is, let's say, 0.5 or 0.9 or whatever the case is, and that has to be measured against a standard graph, because what you'll do with 0.5 and 0.9, you don't know the readings, you want to know the concentration at the end of the day, right? So standards are also given to you, and the standards are also done in the ELISA plate, and that is also measured, where you will get a standard graph, and then eventually you can measure the unknown sample concentration. So basically this is what ELISA is. I hope you understood this. And it would be nice if maybe sometime in your lab, if you know you can actually do it practically, then you'll get a better idea. But yeah, I don't know how flexible they are. Maybe in college. Antibody bound to alkaline phosphatase or peroxidase are used to detect the concentration of the antigen. Like I already said, it's an enzyme linked secondary antibody, let's say, and then a substrate is added and the substrate enzyme reaction takes place. The color changes. Then you can see the amount of your pathogen or whatever the case is, your sample, your antigen that is present. Okay. Oh, so we've come to the end of this video lecture, I guess. But yeah, but to answer your question, it's pretty simple. ELISA is used to detect viruses, but the key reagent is alkaline phosphatase. It's a good question though. To know alkaline phosphatase, you would have to know what ELISA is, right? But I don't think it's mentioned or clearly explained in NCRT. And so yeah, this would be pretty helpful. Yeah. Okay, so we come to the end of this video lecture, and I hope you've enjoyed today's video lecture, and I hope you've learned something new today. And don't forget to like, share, and subscribe to NEET Biology. And I'm available on Facebook, Twitter, Instagram, so you can follow me there for updates. and I'll see you in the next video lecture. Right, so bye.